Okay, so I'm going to talk to you about investing in genomic medicine, but I'm not going to do it from like a market perspective because, I mean, just like everybody, I'm biased to my own perspective, but that's also what I'm most expert on. So I'm going to tell you something about our experience as a startup doing innovation in genomics and supporting genomics medicine and the challenges, well, not one thing is the challenges, but the other thing is what I learned along the way. And hopefully, some of that can be useful in the context of, well, as a case study, how does that apply to other startups? Do they have the same challenges? Do you learn some of the same things? So I moved into, well, why do I have an interest in genomics medicine in the first place? Because I'm actually a computer scientist. You wouldn't know, but I used to make websites and databases. But at some point, I went into bioinformatics, and from bioinformatics into genomics. But today, everything I do is about how to enable genomic medicine to happen, and we're doing it by enabling better data access across different institutions across the world. But how did I get to that point? I'm going to use just a couple of slides to explain to you that background and how that rationale then helps explain where we're at today. So I used to do data analysis for next-generation sequencing back when next-generation sequencing was just introduced into the academic community. So everybody was still using microarrays, and then suddenly there was this new thing. You could put just a sample into the machine, and you get the DNA sequences out, and it was much faster. You could do much higher throughput. This would enable these kind of analysis to scale up, and you'd be able to do a whole genome sequencing, and you could look at not just one gene, but the whole genome. And it was a great time. Well, one thing is, oh, new technology, it's exciting, but all the possibilities that this enables, because not just looking at this particular gene, this particular mouse model, but wait, this actually means that one day this will enable the clinicians even to use genomic medicine for their patients. And once that started to take hold, everybody was talking about, oh, genomic medicine, that's the new thing. You know, when we get to that point, right now, we're just introducing it into the academia, but next thing we'll be introducing next-generation sequencing into the clinic, and then everybody will be able to, when a patient comes in, just sequence their DNA, and you'll be able to give them not just the treatment that's the general treatment that you'll give any patient with that disease, but you'll be able to give the treatment that's customized to whatever's the genomic makeup of that patient. And everybody was talking about how fantastic that was going to be. Okay. I was in the Netherlands at that point, and I was working on specific epigenetic and genetic changes of mouse embryonic stem cells, so very basic research, which would then, I knew from all the conference talks, this would enable genomics medicine somewhere in the future. Meanwhile, while I was in the Netherlands, my mother, back in Denmark, got diagnosed with cancer. And that was like a shock to me. You know, it's always a shock if someone, you know, gets diagnosed with cancer. But then she, of course, went through the treatment. So she went through first an operation, then a radiation again, then another operation, then the first round of chemotherapy, then the second round of chemotherapy. And all of this, while I was looking at what I was doing, I was doing the work that was supposed to enable genomic medicine, and everybody was talking about this was going to be the big new thing. And then, meanwhile, the standard of care for cancer patients had nothing at all to do with personalized medicine, except maybe the operations, because they were quite specific for my mother, right? You had to remove the particular tumor in this particular place. But all the other things, like the radiations, throwing, trying to kill off cells, trying to kill them off with the chemotherapy, all of that is just throwing toxins at the body and hoping that it's the cancer that dies first, not the patient. So I learned a couple of things. One thing was that my research was very far removed from what was a clinical application. I mean, I was looking at individual changes in an individual cell for a mouse, right? And I was making the analysis tools to look at that. And that the standard of care at that point had nothing at all to do with the personalized medicine or genomic medicine that people were talking about in the conferences that I went to for the work that I was doing. And I thought, this is disappointing. And I completely lost interest in that basic research because actually the challenges lie in moving that forward, not just looking at the basic research, but moving it forward into the clinic so we can make this innovation happen. Because already today, we already know cancer is a genetic disease. We already know these are mutations that are driving the cancer, but how can we move that into something that actually makes a difference for the patients who are experiencing it today? So I changed my work from being an academic researcher in basic research and went into an industry and an industry setting. So I actually went to work for Illumina, which you may know is the world-leading provider of the DNA sequencing instruments. And by working with Illumina as a bioinformatician, so the one doing the analysis tools, the software development, computation, I was actually much closer to the clinical application because Illumina, as an innovative company, they had all these ongoing collaborations with hospitals, genetic clinics, where they would get samples from them and they would say, okay, let us show you how you can use genomic medicine for your practice. So I was actually on the team that would receive samples from one collaborator, and we would analyze it the best we could with the latest technology, and then try to show, okay, how that could be used in the clinic. Well, how that could be used in the clinic, that's probably saying too much. But what we were trying to show was how we could see results that could be used to develop diagnostics or actually identify what's the right treatment for the patient. I learned a couple of things. I learned that there was actually some massive bottlenecks in this. By being inside Illumina, I could see data generation was not one of them. I mean, they had machines, and their pipeline of innovation on how to even increase the efficiency and throughput of those machines was amazing. And you've seen today, since now you have the HiSeq X10, and you get millions of genomes of the potential, but the bottlenecks are not in the data generation, it's all in the data interpretation. But why is the data interpretation a bottleneck? If you're just looking at a sequence of letters and you're looking for a change, why is that? What's the difficulty, right? So in my work, I was doing the interpretation, making the tools to do the interpretation. And I quickly found out that it didn't matter what analysis tools I used. You can throw some analysis at the piece of data that we have, and you'd get some result. But what did matter was, did I have any data to compare to validate my results? Because without any data to compare to, a result is just a result. Is it relevant for the clinic? Who knows? Is it significant? Who knows? Is it actually relevant for this particular cancer and it's not just a general change in the population? Who knows? So it turns out that the data interpretation bottleneck is actually, it's caused by the human interpretation required to evaluate the knowledge, but the knowledge is not there, it's not shared, because the knowledge in this context is the data. If you don't have the data, you can't make the interpretation. So for every single project that I had, I would have maybe 10 or 20, or in the best case, we had a couple of hundred samples of a specific disease. But the kind of comparison you need to validate your results, you need thousands of samples. And I didn't have that, and I couldn't get that, although I knew that Illumina was selling machines everywhere, so there's data everywhere. So the point is just to take them, because I'm assuming that the people in this room are not necessarily familiar with the clinical interpretation of genomics data. I'm just going to highlight 3 main use cases. So one is, if you want to discover new genetic correlations, like if you want to be able to say that this variation in the genome is causing this disease or is increasing the risk of getting this disease, you need raw data. It's not until you have all that data and you can compare all of that and you can make a statement, you can draw your conclusions, it's not until that point that you actually have a result that's robust enough for someone to use then later in the clinic, for example, to then test for that mutation and say, yes, this mutation is causing an increased risk of this disease. But in order to make such a discovery, you need data, a lot of data. the second thing is that there's a lot of backlog or history of all the time when genomics has been growing up, and people have, of course, been publishing papers and doing the research all the time with whatever data and knowledge they had available. So you will find in the publication history, you will find conclusions based on maybe 10 samples or one family example of a certain variation causing a disease. Or so they think. It's their hypothesis, and they publish the paper based on that. And frustratingly, a lot of the knowledge that has then been passed on and say, okay, this is something that was found before, okay, so this is the way it is, has been based on publications and knowledge that was insufficiently supported by the data, because the data simply wasn't available back then. So there's a consistent challenge and an effort needed to go back and review and revise what conclusions were made in the past, because when new data becomes available, suddenly you find out, actually, this is not a general a risk factor for this disease. This particular variation is only a risk factor for this disease in the European population. Oh, I see, because those studies back then didn't even have other patients involved in that study. They only had people from the European population. Oh, actually, so that means that we cannot use this result at all if we go to Asia or if we go to Africa. Okay, then you need to gather the data for that particular population in order to make a new conclusion. And this is continuously ongoing. Thirdly, if you want to then use that information to personalize the care of patients, so like taking it one step further, not just saying this variation is causing you increased risk for this disease, but actually going further and saying, based on this mutation, I'm going to give you this treatment because I know with this treatment there's a bigger chance of you recovering, or there's a bigger chance of you manage your symptoms, etc. Then you need not just the genetic data, but you need the sharing of the knowledge of all those cases where that genetic data, the same genetic variation was present, and what treatment they got. So you need to look into the electronic medical records. You need the outcomes of the treatment, and all of that knowledge needs to be pooled and gathered in order to make any new conclusions if you want to standardize what is the treatment, or if you want to make sure that that treatment actually works before you just try something randomly. So in all these cases, you need to be able to gather and aggregate the right data to support the particular hypothesis or the particular study that you want to test. And it's in the order of thousands of data sets that you need, and it's the general, anything that's comparison and validation for whether it's specific disease states, if you want to say whether it's something that's relevant for this cancer rather than that cancer, if you want to say, well, this is actually something that's indication of the disease and not just a variation in a population because you might be biased in your choice of population, or it might be anything that you think might be a bias in your technology. Actually, it's anything that you think, could it be a potential bias in your study design? The only way to rule out that that is actually a bias is by comparing and validating against the data across the different factors of whether it's for this, the way the sample was collected, the way the sample was analyzed, or the way the data was generated. In all of those cases, you need data for validation. And today, although data is available, being generated everywhere, it takes months. It takes months to find out where can you find data that's of the same kind of the type of data that you're interested in right now. It takes months to access it because there's no standardized system for getting the access from different institutions. So you need to go through the bureaucracy that's depending on whichever data collection you want to access. And it's a huge headache. And it's slowing down the research. So when I realized this, while I was working at Illumina, I thought, I can't focus on my job here anymore because this is such a basic problem that is not just causing me a problem to analyze the samples in my project. It's a basic bottleneck for everyone who's doing genetic research across the world. So I founded the charity DNA Digest with the purpose of making it more easy and efficient to access genomic data across institutions, get the community together, rally around a cause, and since it's now, this autumn it will be 3 years ago, we spun out the social enterprise Repositive with the same purpose and mission as a charity, but as a commercial company. Because if you want to create something like this, you need to get a lot of funding if you want to enable a platform that enables the data access, if you want to not just develop the technology, but you want to launch it, bring it to market. And this is what we are currently doing with Repositive, and that's the work I'm doing today. So I'm not the only one who talks about this bottleneck. So it wasn't like I woke up one day, I'm like, oh, this is a bottleneck. No, everybody was talking about it. They were talking about it back then, they're talking about it now. It's like, we need to do global data sharing. The data needs to be available. We can't do genomics-based healthcare if we don't have the data. You get position statements from every clinical, academic, industry group that does research on genomics data, because it's an obvious thing. This is a challenge. We need to address this. So this is not just, it's not just me. It's out there, and it's recognized. It's recognized as a bottleneck. And since then, there's lots of academic consortia, lots of collaborations established on how do we now make standards, how do we get together and make this work. And the problem is only getting bigger. And it's only last year, you would say astronomical, but then last year a new paper came out that was comparing the amount of genomic data that's generated every day, how fast it's going, and how it's going exponentially up. So now we no longer talk about astronomical amounts of data. Now we're talking about genomical amounts of data, because actually the genomic data is growing so much faster. Then you get more genomic data than Twitter and YouTube combined. So it's going further up, and I included the picture here, but I can give you the reference to the paper afterwards. But the problems increase as more data is being generated, because the more data generated is not solving the data accessibility problem. It's just increasing how obvious it is that data is in silos everywhere, and each silo is getting bigger with their own collection of data. So with Repositive, we put together a business proposition, because we want to do this, we need to get investment. Clearly, if we're identifying a bottleneck in an exploding market, that should be a genomical market opportunity for innovation. So we put together a business case where we're not even doing technical innovation. No, we were applying known technologies from web development, data management. We're applying business models that have been successful in other industries, like online data marketplace, the kind of visibility you get. Like today, no one in this room would go to each individual airline to find out which of them has a flight to Rome on the 20th of June, right? I mean, you wouldn't do that. You rely on tools being available that make the data easily accessible, that gives you the information and the knowledge you'd need to make a decision. So in those kind of industries, that kind of information and knowledge availability is already implemented, and you're using it because that's the default. That's what you expect. But that is not the case when it comes to research. Streamlining access to scarce resources. That is seen in many industries as well, including healthcare, actually. So there's already been several startups, there's actually also in UK and US, but in UK there's Antidote, for example, that make it easier for pharma companies to recruit patients for their trials, because that's difficult. Okay, so we have a startup that's doing that, and it's streamlining the access, it's helping everybody, making the research happen faster. And we're using the business model of having a freemium community so that we have a network effect happening where you get the engagement of the whole community, not only of those that are paying customers. And all of these are known business models and known technologies. We're not even inventing something new. So it should be a good business proposition. It actually turned out to be quite difficult. So in the very early stage of a startup company, you go to angel investors and you find the individuals who believe in your cause, and they each invest small amounts, and then you can get the investment together by talking to several of those individuals. But now we've gone to a stage where we need venture capital funding in order to grow further. And we managed to close a funding round of Series A of 2.5 million. But it was not easy to do. And it took a long time. And I'm going to point out some key issues there, because I think they're relevant also for other innovation in healthcare or in the space, whether it's genomic or other healthcare. So by us making a genomic data community marketplace, how do you categorize that? So we would go to a venture capitalist who invests in software, right? Invest in software and technologies, even doing maybe healthcare apps. And they would look at us and they say, oh yeah, you're making software, but I mean, you don't look like one of our software companies, because you're clearly doing something sciencey, something very, you should probably go talk to the biotech guys over there, because they know about your space, they'd understand this. Okay? Go to the biotech guys over here, and that invest in drug discovery companies and in lab technologies, and they look at what we're doing and they're like, what is software as a service? They ask us, and we go like, okay. It's going to take a long time, this conversation, right? If they don't understand the way that we're delivering the product to market, it's going to be a difficult conversation. So we found that the investors that we were talking to, although they're all, of course, invest in innovation, right? They want to invest in the latest, the greatest. But the difficulty comes if you're in between two chairs, in between two different segments, market segments, and you don't fall straightly into one of them. It's very difficult to find those lead investors who are willing to step up and invest, say, yes, this is a great idea, see how we can help you, and we're willing to put our money behind this, and then go and invest there. Because none of them feel comfortable outside their own space. So the way that our Series A came together, it took a long time, but eventually we found in the conversations with the investors, going back and forth, like, oh, so you're not comfortable with software, oh, so you're not comfortable with genomics, oh, how do we sort this out? How do we get the investment round together? We ended up actually having 3 investors, each investing at the same level of funding, one in tech, one in, tech and IT and software, one in biotech, and an angel investor, a super angel who has an expertise in biotech and marketplaces. So that special combination took a lot of work. And we had to handpick the specialists subsequently, because since none of our large investors actually had the knowledge of our particular area, we had to go handpick the particular specialist investors who had the knowledge about genomics, or knowledge about genomic software, or about bioinformatics, to get them on board as individual angels to make sure we had that right knowledge of the space on board that we need to grow as a company. And I think, but maybe other people in the room can give some feedback on it, but I think that's a common situation. So if you ask me, what can we do to make innovation happen faster? I think we have to realize that if you do innovation within a discipline, you only get the incremental innovation. But if you want to make those really step changes, if you want to do that radical innovation that is moving borders, that's changing what is possible, you're actually going in between disciplines. But if innovation is funded by investors who are looking within their silos of disciplines and only investing within their particular comfort zone of particular area that they invest in, how are those innovative cross-disciplinary things going to get funded? So my two points about how to make innovation happen faster is identify those bottlenecks and invest across disciplines. I think that was my last slide. So thank you for listening, and I'm happy to tell you more about everything we do. And if there's not time in the questions, we can talk in the break. Thanks.
Thank you very much.